Hey everybody, it's Nico here with a lecture on meiosis. If any of you remember the old song, Let's Talk About Sex by Salt and Peppa, that might be what they were talking about because meiosis is all about making gametes, which are eggs if you're a biological woman, chromosomal woman, or sperm if you're not. So we're going to talk about how to make egg and sperm today using meiosis. And we'll also talk about how those processes are similar across both egg and sperm. And we'll talk about how they're different for egg and sperm. And I'll give a couple nods to how this relates to mitosis to reinforce that concept that we all know we learned back in the day, middle school, high school, maybe we forgot it a lot. So we'll get back to it. So meiosis, we're talking about sex, we're making egg and sperm. Mitosis, we are talking about not sex, we are making the rest of the body cells. Okay, let's get started. So this is essentially the process that germline cells go through in order to create gametes, egg or sperm, which are haploid or have half the number of chromosomes. So for humans, half of the number of chromosomes is half of 46, which is 23, so egg and sperm both give 23 chromosomes. We want to give half of our set to our children so that our partner can give the other half of the set, or our donor, whatever the case may be. And so whatever we have, we just cut it in half. So we're humans, 46, half is 23, but other animals have other numbers of chromosomes. So when they go through meiosis, if you have 40 to start, you'd have 20 when you finish meiosis. Not 42 start, 40, there we go, 40 to start. Half of that is 20. Likewise, if you had 36 to start, then you'd have 18. Okay. So the process of meiosis first starts with a round of DNA replication. So we take our chromosomes, which are condensed now and linear, and we replicate them in S phase of the cell cycle to make a X-shaped chromosome with two sister chromatids attached. This is followed by 2 rounds of cell division as compared to 1 in mitosis. So we take 46 chromatids and we turn them into 92 chromatids that are attached together. And then we separate those twice, resulting in 23. So 46 chromatids to 92 chromatids, back to 46, and then to 23. And there'll be diagrams later, don't worry. Just setting the stage, because again, this is supposed to be a review. You should have already learned this, we're just brushing it off. So crossing over is a process that happens in homologous chromosomes. And remember that homologous chromosomes are chromosomes that are the same, and we got one from one parent and one from the other parent. So chromosome 1 from your mom is homologous to chromosome 1 from your dad. Your X chromosome from your mom is homologous to your X chromosome from your dad if you're female. And then so on and so forth. X and Y are functionally homologous. We'll get to that later. It's another lesson for another day. But it's the same type of chromosome, essentially. So we see switching of information between them. And now, close your eyes and envision this. If you're receiving a chromosome and it has information that's been switched to it across its like pair, chromosome 13 with chromosome 13, swapping material, whose combined information are you getting? If you are the carrier of that combined chromosomal information that's been crossed over, you'd be getting let's say you got this from your mom, so it'd be your maternal grandmother and your maternal grandfather's chromosomal DNA mixed together. And This is a good thing, and it creates genetic diversity. It makes novel combinations that, in theory, enhance our potential to survive as a species. Hopefully, fingers crossed. Um, so if you've ever looked at your parents and wondered, how am I just so much better than you and grandma and grandpa on both sides, it could be crossing over there. So how crossing over works is our two homologous chromosomes, which, since they're the same type of chromosome, they would be the same size, same shape, but we got one from one parent and one from the other parent. That's what the colors denote. And since we've got, all these genes along our chromosomes, and then genes can have different versions, which we call alleles. Those are typically depicted with letters, but sometimes numbers, and a lot of times we use capital and lowercase to distinguish between the two types. And so here we see, let's say this chromosome in navy here came from mom, and she has a big A, a big B, and a big C chromosome. And then the one that we got from dad in light blue has a little a, little b, and little c. So if big A, big B, and big C are dominant, and little a, little b, and little c are recessive, typically if crossing over didn't happen, which sometimes it doesn't always happen, it just sometimes happens. So you could inherit the dark blue chromosome, or you could inherit the light blue chromosome. But you could have a little swappy when these chromosomes find each other during the cell cycle. When they're lined up, they have a chance to kiss and exchange some information, basically. And then we have a chromosome that has the big A, big B, but the little c, which was not a chromosome that either of the cell maker's parents had. So now, instead of your mom giving you either her mom's or her dad's copy, she's giving you a copy that's either mostly her mom's with a little bit of her dad's, or mostly her dad's with a little bit of her mom's. And again, this only sometimes happens, and it's a good thing. It, generally just creates new phenotypes that we haven't seen before. So gametogenesis is the term for creating egg and sperm, because there are gametes, right? And genesis means to make or to originate. So we're making egg and sperm with gametogenesis. In terms of egg, it's a really, really big cell. It's literally the largest human cell in the body. And we said before that meiosis creates haploid cells, which have half the number of chromosomes. So this n equals 23. That's a human-specific number. If you were something with, 14 chromosomes, half of it would be 7. So whatever is half for you, that's what mom and dad will both give. Mom also gives something called mitochondrial DNA. So inside your mitochondria, which again, think all the way back to your early science classes, mitochondria are the powerhouse of the cell. They help to create cellular energy in the form of ATP, and they're very important, but inside of them they have their own little source of genetic information. It's a circular chromosome. It's called mitochondrial DNA, and we get it from mom. There are some caveats here. Wherever there's a rule, there always seems to be a scientific exception, but typically the vast majority of people's mitochondrial DNA is maternally inherited. So maybe put a little asterisk there. And ultimately we make an egg in the end. Oops. And we'll have a diagram in a little bit. This is not something that I would ever quiz you on, the gory details, so that's why this is a very low diagram PowerPoint. So the first thing we do is we do our replicative division. So replicate means to copy or double, right? So we go through DNA replication, make a second set of DNA, which occurs during S phase of the cell cycle. And then we divide the first time. So this is again something that I would never make you memorize the gory details of, but this 2n, 2n, n just represents that we're having it at a certain point. I honestly do not care if you know when it's a primary oocyte or a secondary oocyte. just know that it's a process and there's lots of names for the things along the way. Then we go through the second round, which is equational. This is all I would focus on, what's written here. So after we've replicated and then divided once, then we divide a second time to get our half number of chromosomes that we need. And in women, egg makers, I guess I should say, we make one ovum, and then because eggs are so big, part of that is because they need a lot of cytoplasm. And so even though we divide something in half twice, which would make 4 things, with eggs, we only divide something in half twice. We do it twice, but 3 of them just end up getting basically none of the cytoplasm. And sometimes a couple of them don't even exist. So we get 1, 2, or 3 polar bodies, which are non-functional, non-viable, do not make a baby. You cannot fertilize a polar body. And then 1 viable egg that could be fertilized. In terms of sperm, for our sperm makers, this is a smaller, more vulnerable cell. I'm sure you probably remember learning about, the fact that we need a lot of sperm in order to fertilize an egg typically. These are typically, they have mitochondria, and as I mentioned before, there have been exceptions in terms of mitochondrial DNA inheritance, but typically when we talk about making sperm, we focus on the nuclear DNA contribution because that is really like the main one. So we make sperm using spermatogenesis, we make eggs using oogenesis, and both spermatogenesis and oogenesis are a form of gametogenesis. Does that make sense? Hopefully. So you can do gametogenesis 2 ways. Spermatogenesis if you're a sperm maker, or oogenesis if you're an egg maker. So here's a diagram, again, with our little ends. So in this case, we take something, again, and we do a replicative division. So double it, copy the DNA in S phase, divide it in half, and then divide it in half again. And this time, dividing one thing in half twice does result in 4. And so we get 4 viable sperm cells, which is good because we need a lot of sperm to fertilize 1 egg. So very efficient process. All the sperm kind of split up the cytoplasm and things equally. So very efficient, and you always, you get 4 versus 1 egg and 1, 2, or 3 polar bodies. Then the magic happens. The unification process occurs. This results in, the 2 haploid sets coming together in the nucleus, forming a diploid organism with 46 chromosomes if that organism is a human, and some other amount if they're not. So this is a video for you to watch on your own. If you feel like some additional content on meiosis would be good, if you feel like looking at those diagrams that I will never, ever, ever, ever quiz you on, if you feel like you want to see those, check out this video, do some other sleuthing online. There are lots of people confused about meiosis, and so I'm sure you can find some resources. I would also recommend the Amoeba Sisters. So just to recap, meiosis is a 2-step process with a replicative division and then an equational division where we don't replicate again, which is different from mitosis, which is a 1-step process. In mitosis, we always take a cell with the full set of chromosomes and make more cells with the full set of chromosomes, whereas in meiosis, because of this replication and then extra equational division step, we end up having the number, so we make haploid cells as opposed to diploid cells. Gametogenesis makes egg or sperm via spermatogenesis and oogenesis. And crossing over can occur during meiosis, which is generally a good thing and produces novel genetic combinations. And it could be why you have your maternal grandfather's freckles, but your maternal grandmother's sparkling green eyes, or whatever the case may be. Anyway, that's about it for meiosis. I hope you learned something. Let me know if you're confused. Thanks, everybody.